Yeah, all right, then. So I'm going to resume. So our next speaker are going to be Ard Louis and Kamel Dingle, who will be talking why the word is simple and then the AIT coding theorem in machine learning and biological evolution.
Okay, hello everybody. Oops. Yeah, welcome back after the break. So just a bit of a background for myself, just before I jump into the talk. So I've been interested in algorithmic information theory for like 10, 11 years or so when I started my PhD with Ard Louis over there. And so we, since that time, I've tried a few other different areas, and I'm now kind of with discussions with Boumediene, who has a background in machine learning. We've been starting to talk about some of the kind of overlap of some of these ideas. I'm more interested, or my background at least, is more interested in applications in biology, science, and things like that. And now I'm taking a sabbatical. I've got a small essay here of associations, because I'm taking a sabbatical from my main university. So I'm associate professor in the Gulf University in Kuwait, but I'm taking a sabbatical. I'm doing some research now in Cambridge University in theoretical biology. I'm using AIT in biology there, extending some other work which Ard will mention. And I'm also visiting associate at Caltech, where Boumediene and Professor Houman Ohadi, I'm trying to get into time series and seeing if I can find some AIT applications and other kind of investigations there. Okay, so this talk will be split between me and Ard. I'll start and Ard will carry on. Let's see, how do I? Yeah, okay, great. So my main interest here is that in this talk is connection between probability and complexity. So Marcus obviously has been talking about Solomonov induction and algorithmic probability, the idea that probability and complexity are intimately related. His interest has been mainly focusing on induction and inferring what's going to happen next. I'm interested in a slightly different question, which is to say, can I just look at some structure, some pattern, let's say in nature, maybe a molecule or some other kind of pattern like a tree shape or maybe a time series, just looking at the object itself and estimating its complexity, can I use that to infer or make some guesses about what the probability of that object is? Okay, so it sounds a bit of magic, and actually it's been described as magic, this kind of distribution, that you can make an estimate of probability without looking at historical data, because obviously machine learning and traditional statistics is you look at data, count frequencies, make some estimates of probabilities. This kind of theory promises perhaps to be able to make some kind of a priori probability predictions without actually looking at historical data. So just go over this very quickly because it was already described. So we've got the so Levin proved this coding theorem. Basically says if you have a computer, a universal Turing machine running a random program, the probability of outputting some string X or some pattern X is given by 2 to the minus K. So this is Kolmogorov complexity. Obviously, we were aware of that. So that's what it is in theory, and this kind of algorithmic probability has been used in different kind of philosophical contexts, perhaps induction, applied in other areas. But if you think about it kind of carefully, trying to apply an actual practical setting, like what's the probability of this biomolecule or what's the probability of this particular time series, there's quite a few difficulties in directly applying it. One of them is that K is uncomputable, as you will be aware. Another is that it's assuming a universal Turing machine, which is not obvious that exists just sitting around in the natural world. And there's a random program, which again is not obvious that there's actually technically random programs in a relevant field. It's an asymptotic theory. You have these order 1 terms, machine dependence. Again, it's not making precise predictions. It's more like a kind of a like scaling arguments. As complexity approaches infinity, you can make these kind of relations. So on the one hand, we have this very promising result, which seems to make a striking claim that complexity and probability are connected. On the other hand, the theory is kind of rather abstract. So what I started to do in my PhD, and we've worked on in the 10 or so years since then, is trying to see if we can bring that down into the kind of concrete world. So looking at a computable distribution, imagine we have a computable distribution, meaning like a distribution you can actually calculate over, let's say, for example, like biological structures, like you have a bunch of different structures, different shapes, like protein shapes or RNA shapes. If you have that probability, you can make, is well known from Shannon information theory, you can make a prefix-free description using log 1 over p bits. So you can describe or identify any of those objects x with a code of that length. Then what you can do is you can just use that as an upper bound on the combinatorial complexity. So combinatorial complexity is supposed to be the shortest description of some object. And so if you have a particular description based on probability, that would be an upper bound. Here the f and the n. So f is the function that you're investigating. So let's say you're trying to calculate, you've got DNA sequences and the function is telling you what the shape of the molecule is going to be. Or maybe let's say you've got an ordinary differential equation system, you've got a bunch of parameters and the function is the equation system itself, which is going to tell you what the solution is. Okay, so f is some function that is producing outputs given inputs. And n is just a parameter telling you like how big your system is. So what we got here is just if we can rearrange the equations here, and you get this upper bound on the computable probability p, but you've still got an uncomputable complexity there, and this function and this n, and order 1 term. So again, we can't quite apply it in the practical real world. So we make some approximations. So one thing we could say is, if we restrict ourselves to functions which are simple functions, technically order 1 complexity, then we can make an approximation that the combinatorial complexity is going to be basic. Well, actually, that's not an approximation. That's actually a technically correct relation. So if you have simple functions, you can make that approximation. We can make an approximation or a kind of a hopeful approximation that the value of n that you're inputting there, meaning the size of the system that you're using, the length of your DNA sequences, is not going to be changing the complexity too much, which is kind of reasonable. And then a sort of a requirement rather than or an assumption that your map that is producing your outputs is going to be somehow nice. By nice, I mean things like it's not going to produce pseudo-random outputs. So for example, if your map, you give it a random DNA sequence and it produces, let's say, pi, the digits of pi with high probability, typical Shannon entropy complexity measure or a Lempel-Ziv or a CTW measure will not be able to detect that kind of complexity, and so you won't be able to handle that system. So we're just going to assume or restrict our attention to maps for which are kind of nice in an unspecified sort of sense, for which we hope that real-world compressors like Lempel-Ziv will actually approximate the combinatorial complexity. Paul Vitani actually has argued that this is quite reasonable in many real-world settings because, in the real world, many systems are not UTMs themselves, and so, they're unlikely to be producing these pi or kind of crazy things themselves, in which case you're unlikely to be missing important details. Okay, so with those approximations, and a couple of other arguments that we came up, we present this kind of upper bound here. So K tilde is some everyday kind of complexity measure, like we were using the 1976 Lempel-Ziv complexity measure, and also a bunch of other ones. But you can use other complexity measures, and we get this kind of upper bound relation. Okay, so just looking a bit more at this, and we've got these two values, A and B. So A and B are kind of considered kind of fudge parameters because we're making all these approximations, but they're not completely free. They're not just completely tunable. A is actually a value which you can often predict quite accurately just from knowing the total number of different patterns or outputs that your map can produce. And in fact, A will be basically close to 1 if you scale things correctly. B is typically, we assume this to be 0 most of the time. We argued why that would be reasonable in the paper, but it's not always 0. Sometimes you might have to fit that kind of bound. So what have we come up with here? The summary is that we're saying if you have some kind of a system like DNA sequences to a biological structure, or maybe differential equation parameters or some kind of solution, we should be able to predict an upper bound on the probability of the patterns just using the complexity estimate and maybe some estimate of the A and B. So why is that nice? It's kind of like being able to predict the future in a sense, as say, just show me the pattern without looking at historical data. I can give you some estimate or at least an upper bound on how likely it is to appear. So we tried to do this in practice then. Oh, sorry, there's a couple of other points that we found in this paper that when we actually study this for a whole bunch of systems, I'll show you some soon, many of the output patterns will actually have probability far below that bound, which is interesting. But also interesting and perhaps more surprising is that with high probability, you're likely to produce an output which is actually close to that bound. Okay, so that sounds like I'm saying two opposite things there, but they're not quite the opposite. Basically, most of the probability mass is associated to outputs which are close to that upper bound, but many of the outputs themselves actually have low probability. Okay, so let's have a go. This is what we did. We tried this on a bunch of real world systems. So again, I wanted to bring AIT into the concrete real world of actual predicting actual, probabilities for real systems. This was taken from that paper. We looked at RNA, computationally generated RNA, ordinary differential equations, a financial model, Ornstein-Uhlenbeck system, L systems down here, model of plant development, and some matrices. I'll focus just on the one down in the left-hand side here because it's the easiest to understand. So L systems is it was a kind of a system that was generated for making computationally generated plants. I think they wanted to use it maybe in like computer graphics or something. So it has a kind of a little mini algorithm that you can encode, and it does things like grow, branch, grow, etc. Yeah, so you can randomly sample these instructions in the L systems formula, and then it will implement those instructions and grow a certain plant. Okay, shape. So what we got here is each of these dots is a different plant shape. It just highlighted the two examples of interesting ones. So at the top left there, you've got this small, fairly simple little plant shape, which has got a high probability and a low complexity. And then also illustrated down the bottom there, you've got this irregular, complex, asymmetric kind of crazy looking plant, which has got, low probability and high complexity. And each of the other dots are also some other different plant. So the black line is the predicted upper bound probability. As you can see, the data follows the upper bound reasonably well, a couple of violators. But basically, what we're saying here is just from looking at the actual plant structures themselves, you can make a pretty good upper bound estimate of the probability, which works reasonably well. So this we called a simplicity bias, So there's a bias for simplicity here in the sense that simple things are more likely than complex things. But unlike the original coding theorem of Levin, in all these maps you have these low complexity, low probability points down here. I've just written a paper on that. It hasn't appeared in the world yet, but it will do soon. I'll just skip over. Okay, I'll go over this bit quite quickly because Ard needs to continue. Recently with Boumediene, we have had a look at seeing if we could see simplicity bias in natural time series. So we looked at the World Bank open data. They've got a bunch of data on all kind of things like percentage of people with access to renewable energy and number of people with HIV in Germany and all kinds of random data. We looked at these time series data. We did a discretization process. So here is an example of this is real data for some HIV cases in Germany. Over there, you can see that the number of HIV cases is sort of wiggling up and down. We did a discretization process, turn this into a binary string so we can more easily calculate its complexity and probability. So the process we did here is pretty straightforward. We just said, if your value is above the mean, then you're yellow and you're going to be given a 1. If your value is, if the series value is below the mean, then it's going to be blue with a value with a number 0. So this becomes 1 1 1 1 0 0 0 1 1 1 0 0, etc. Yeah. So this is the binary string that corresponds to that series there. So in that database, for the kind of time frame we were looking at, there was maybe 20,000 or so of these series. For each of them, we convert them to a binary string, calculate their complexity, and then to see if there is a relationship between complexity and probability. So what we found is this graph here, which is quite a nice graph. So the black line is again the upper bound prediction. Each of those dots is a different binary representation of a time series. Obviously, it's not a perfect upper bound. There are several points here that are above the bound. Presumably, they are somehow very kind of popular or somehow time series patterns that are somehow overrepresented in the real world. And there's a few points down there also above the bound. But generally, you can see the same kind of pattern there of simplicity bias in these time series. Very quickly, just going to skim over this in 20 seconds. We also looked at, in a different paper, trying to look at the lower bound on the probability using the complexity of the output structures themselves, so the complexity of the patterns, as well as the complexity of the input programs that make that pattern. So that was the bound that we generated. We tried to use it in practice also in a couple of different systems. So we're looking at RNA here, and finite state transducer, and a perceptron model from machine learning. So for the RNA, just using the complexity of the output, we have this upper bound, which is okay, but not that great at predicting probabilities because there are so many points that are very far away from that bound. By including the complexity of the inputs that make that output shape, we managed to improve the prediction of probability a lot for RNA. For the finite state transducer, we somewhat improved it. It wasn't perfect, but there's definitely something going on there. For the perceptron, it was less clear. There was a very mild improvement. So I'll let Ard now continue with more applications in perhaps biology, machine learning, and we'll take questions at the end.
You can also myself and how's that? Is that good? Thank you very much. So the way I got into AIT is that I had been working on some models in evolution for a little while, and I kept seeing this bias towards simplicity. I had Kamal as a PhD student, came from a mathematics background, and I said, find something that explains this. And we tried lots of different things. Then someday Kamal came to me and said, look, there's this coding theorem. It predicts this. And then we realized the problem, of course, was that we were looking at RNA models. All the beautiful properties of the coding theorem depend in subtle ways on the fact that these are universal Turing machines. You can actually show that it's very hard to prove the coding theorem without using you need the halting problem to make it work. And so then, being physicists, I said, well, okay, I can't prove it at this level, but it's such a strong prediction. It must also hold for weaker computable devices. And so this is kind of how we approach it. And this is kind of a typical for the mathematicians in this room. This can be a horror. Physicists can horrify you. Sometimes we just try something and then we'll try it in practice and see if it works. And there are many examples. Very famously, the epsilon expansion in renormalization group, incredibly powerful. You can show its radius convergence is next to 0, but we use it for several. We go way well outside of its bounds, and it still works. And that is the kind of way I'm going to approach this here. I'm going to look at two questions. One is basically some fundamental questions in machine learning, and then some questions in evolution. There we go. So physicists are always very suspicious of any kind of thing that has too many parameters. Famous story by one of our great heroes, Dyson. Went to talk to Enrico Fermi about a theory he had, and Fermi said, well, how many parameters do you have? And Dyson said 5. So my friend John von Neumann says, with 4 parameters, I can make an elephant. With 5, I can make a Elizabeth's truck. So we're saying that, if you have too many parameters, it's not going to work. And in machine learning, this intuition has been formalized in what people call bias variance trade-off. I saw a talk recently by at Cornell, which is where the talk on machine learning, where the speaker said bias variance trade-off separates the adults from the children in machine learning. The idea being you make your model too complex, too simple, you can't fit the data. You've got bias, you make your model too complex, and you have the problem that you overfit. And so the claim in this very recent lecture on machine learning at Cornell was, if you want to understand machine learning, you've got to find this kind of balance between these two. Now, the problem in this kind of deep learning revolution, I showed this quote, it's a very famous paper quote from Alan Turing. We use neural networks, and neural networks have huge numbers of parameters, millions, billions, and the more recent, all this on these more modern models have extraordinary numbers of parameters. So they're well outside of the make the elephant's trunk wiggle, von Neumann's quote. And the question really is, why do they work? Another way of thinking about this is in this more kind of slightly more formalized way, Bayes variance trade-off, where you're not counting parameters, but you're looking at something like the capacity of the model. How many functions can the model describe? That's the formalized way of doing it. Well, we know that these neural networks are highly expressive, almost perfectly expressive. That means that they can basically express any function that you can think of. This became very popular in the kind of neural network literature with this paper by Zhang et al., Understanding Machine Learning, Deep Learning Requires Rethinking Generalization. I just looked it up, it's 4,000 citations in the last few years. And what they basically did is took an image, this set, here's called CFAR10, sorry, randomized labels, so cat became dog, vice versa, and showed that a machine learning algorithm could, if you gave it that data, could perfectly reproduce that data. In other words, it could learn this very complicated function with random labels unlinked really to any property of the images. And the question then was, if a neural network can be trained to zero training error on arbitrarily complex data, how come it, when I give it data that has something that, how come it doesn't do that when I give it data with correct labels? Why does it learn rather than memorize is basically what they popularized. But it's, in some sense, it's obvious that neural networks could do this because they're extremely highly expressive. So of course they could express that. The question, I think the real thing this paper showed us was not that they could do this because we knew that they could. It showed you that SGD was an efficient optimizer. You could find these solutions quickly. It's a slightly different question. The question is much older. And in fact, here's another example of it that we've just done just to illustrate it. So here I've got some, dots. I fit the polynomials to it. I fit a lower polynomial. I get some bias. Fifth order can't fit it. I fit a 20-order polynomial. I get variance. It starts wobbling around. And actually, this green line and the orange line and the blue line, which are all overlapped to each other, are basically some very simple 1,000 width fully connected networks, and they give this very smooth curve. So the central theoretical conundrum of neural networks really is, why do they generalize at all? Why did, given that this neural network can produce any polynomial, any function, it could have made any of those functions fit the data. I give the data, I don't tell it anything else, and it gives me the smooth function. Why does it do that? That's a really interesting question. I think it's one of the kind of central conundrums of the neural network field. And it's an old one. So here's a very famous paper by Leo Breiman, statistician, very famous statistician, random forest, one thing he discovered, for example, or invented. And in 1995, he wrote a paper about his many years of refereeing for what was then called NIPS, now NeurIPS. And he said his first question was, why don't heavily overparameterized neural networks fit the data? So this question is actually extremely old and is very interesting. Why don't they overfit? And I wanted to distinguish two questions because my experience with referees is that they find in this field they find it tremendously hard to do so, which is, first question is, why don't overparameterized neural networks fit? Why don't they overfit? Why don't they just give you randomly wobbly functions? It's actually true for a wide range of overparameterized systems. And then there's another question which I think engineers care about, which is, given that my neural network generalizes well already, doesn't suffer from this overparameterization problem, can I figure out some way of tweaking it or doing something to make it slightly better? That's a super important question, right? It's a different question from the fundamental one. And I don't think you're going to properly understand question 2 unless you understand question 1. So I'm only going to look at question 1. I just want to make that really clear because my experience is that talking to machine learning people is that these two are conflated. And so one way that question has been popularized recently is by looking at this Bayes variance trade-off and pointing out if you keep increasing the number of parameters or the capacity of your model, what you find is that a lot of models start, they initially follow Bayes variance trade-off, and then they have this double descent. As you make them more complex, the error goes down. Again, it's not surprising. We knew this is true because highly overparameterized neural networks generalize well. So we knew that if you make them very big, they would have this kind of property. I've worked on this a little bit. If you're interested in the technicalities of it with let's say, looking in the Gaussian process limit, we can do this as well. But I think the real question is really, why is this low? Not why is it double descent. I don't think double descent is actually that deep or even that interesting. I think the interesting point is not why does it go up and then back down again. The question is, why does it go down here in the first place? So I'm going to give a very simple model problem. Kind of physics like simple problem. So I'm going to look at learning a Boolean function. So let's assume that I am a doctor and I'm doing some triage on COVID. So you come to me and I've got symptoms. I ask you symptoms. I ask you 7 questions. You have a fever, you have a cough, you lost sense of smell, are you older? You have comorbidities. And then the question is, should I send you to the hospital or not? Because that's a question which costs some resources that may be limited. So if you have all these things, I'll send you to the hospital. If you have all of them, but you're young, but you have comorbidities, I might send you to the hospital. If you have the symptoms, you're young, but you have no comorbidities, I might say, sorry, mate, stick it out at home, etc. So the question then is, if I have a doctor like this who makes these kinds of answers, if I give you a few of the answers, can you predict the rest of the answers? Well, intuitively, you think I probably could, right, in this particular case. But can you do this in a more generic sense? So if I have 7 questions, I've got 128 different possible sets of answers. And so for every one of those 128 sets of answers, I've got a send you to the hospital or not. So that's one function. One Boolean function is given all those 128 answers to send you to the hospital or not. And I'll call yes, 1, and no, 0. So then there's a very simple functions, like I send everybody to the hospital, all 1s, or I send nobody to the hospital, all 0s, right? Those are probably very easy to learn. And the question is, if I give you a certain number of examples, can I learn the rest of them? This is a nice model problem. And what I'm going to do for this particular model problem, and then argue that this holds more generally, is that one way of thinking about this is to think in terms of the mapping of a neural network from parameters to functions. So neural networks are parameter function maps. You find some way of picking parameters, and then it's effectively a function which takes inputs and then gives you outputs, right? In this case, it's a simple one. I give you a bunch of answers to these questions, and it tells you the Boolean function that comes out on the other end. And this is a useful way of thinking about it, because I can then ask myself questions like this. If I randomly pick parameters, how likely am I to get a certain function? Right? So there's 128 possible answers. There's 2 to the 128 possible functions that I could have, 2 to the 128 different ways that the doctor could answer those questions. That's 10 to the 38, a very large number of functions. So you might naively assume if I just randomly pick parameters, I should get any given function with a probability of 1 in 2 to the minus 128. That's not true. So with my student Guillermo, we just looked at this. So here I have the probability ranked for the first 10 to the 8 functions, 10 to 7 functions. We just randomly picked parameters and saw what does the neural network do. And what you see is it follows a nice Zipfian law. These little differences are for different kinds of ways of initializing the parameters, because obviously you want to have some measure on your parameters. So some Gaussian measures typically taken. And first thing to note is that these probabilities are high. Okay, so not 10 to the minus 38, but they're like 10 to the minus 1, 10 to the minus 5. Those are extremely high probabilities compared to the average probability, which is 10 to the minus 38. Right? So there's some set of functions that are much more likely to be found than others. So which functions are they? Well, from Kamal's talk, you would have probably guessed these are the functions that have low Kolmogorov complexity. So you can take that output function, zip it, or use some other measure. There's many different measures you can use. They don't really matter that much. And here I show you that the most likely function, in fact, here is with a very high probability. This is the function of all 0s or all 1s. It's the trivial function. Right? It has also very low complexity. And then this is this upper bound that we have, and it fits the data extremely nicely. And so what you see is this neural network, upon random sampling of parameters, is exponentially more likely to pick simple functions rather than complex functions, which is what you expect from this kind of coding theorem, Solomonoff type of ideas. So that's interesting, right? These neural networks are extremely strongly biased towards a small subset of the space of all functions. You can prove this for perceptron. We have an exact proof that you can, I'll skip this actually. If you are interested in it, you can look up some papers where we have a, sorry, I'm hearing something. Is this me or no? Okay, no, fine. Oh, I thought I did something wrong, sorry. Yeah, so I've got here a paper where we've proven some partial results for perceptrons. You can prove some nice exact results. Perceptrons are nice and easy to work with. The minute you make neural networks, it gets a lot harder. But we think we can prove analytically that there is also a biased reciprocity in these Boolean problems. So just to remind you what I've done already here implicitly, I can generalize to more generally. So I'm going to think about neural networks as function approximators, and I'm going to look at post-thresholded functions. So functions where if I'm looking at image data, for example, if I give you these 5 MNIST images, a function is simply going to be, given these images, what does the neural network do? What labels does it predict? Right? So if it predicts 50419, it gives you 0 errors. If it makes one of these incorrect, it confuses this one with a 7, it's got 1 error, etc. So I can look at all possible functions of this type. It's slightly easier than looking at the pre-thresholded versions. And then if I look at things this way, lots of things become very easy for neural networks. I can write down a Bayesian kind of approximation, a Bayesian approach. I look at the posterior. How likely is my neural network going to give you a certain function if I condition it on a training set S? Okay, so I've got some training set, which is a bunch of images, say a bunch of MNIST images. Well, it's just Bayes' theorem, right? So I've got a prior, and I've got my posterior, I've got my likelihood and my marginal likelihood. And the nice thing about this is my likelihood is extremely simple. If I want to train to zero training error, so I want to have always correct, then basically my likelihood is the likelihood of a certain data set given a function is 1 if that function gives me correct, is correct on the training set, and 0 otherwise. And therefore my posterior and my prior are directly proportional to each other. And therefore any bias towards simplicity that's there in my prior, which I just calculated for you, is also going to be there in my posterior. They're going to be directly proportional to each other. And you can do a lot more with this Bayesian approach. It's like a Bayesian one-on-one, and it's very simple. Now, I tell you that this neural network is biased for simplicity, and that's interesting, but is that really do something for us? So I'd like to see if I could break that bias for simplicity. And interesting, neural networks have, in certain cases, transitions to what's called chaotic behavior. So if you don't use ReLU activation functions, but say Tench or error function ones, then you can show that if you initialize with very wide initial variance of your parameters, you enter a chaotic regime where in the limit of infinite depth and actually relatively wide width, you go to a fixed point where effectively the neural network, you can prove for Boolean functions, will pick a function at random. So there in that limit, it will pick a function with 1 in 10 to, if a function will have a probability of 2 to the minus 128 of finding, so 10 to the minus 38, extremely small probability of getting it. That's the opposite limit of the simplicity bias, where I pick a function at random. And that's nice because I can, if I'm not quite in that limit, but I start moving towards that limit by, for example, making my initial parameterization wider, should start seeing a breakdown of simplicity bias, right? Because that's the opposite of simplicity bias. It's a uniform sampling over functions. And so what we then did is what you see here, if I make the width wider and wider and wider, or make it deeper and deeper and deeper, both are 2 ways of going towards the chaotic regime, then Occam, who you see here, slowly disappears away, goes away. So less biased, less Occam. And here you can see it again. So here's that prior I showed you just ranked. So how likely are the functions? Here's the prior, but now look at the Lempel-Ziv, and the different colors are more down. As it goes down, there's more and more into the chaotic regime. And what I'll show you here is basically how well does a learner learn on different kinds of functions that I've trained on. So there's 128 function outputs, so I'm going to train on 64 of those. And I'll see if I give you 64 answers, how likely is the doctor, can I predict the doctor can do on the other 64? And so on the x-axis here, I've got different target functions, which are basically how complex is the doctor. And then what I have here in this blue line is what a neural network does. And you see that neural network's error is good for simple functions, and eventually gets bad for complex functions. Here, this red one here is a slightly chaotic, not very chaotic, but slightly chaotic neural network. But I've pushed myself away from that nice simplicity bias I had to something that's less simplicity biased. Okay, and already quite quickly, my generalization performance deteriorates dramatically. You see that even on simple functions, this thing really can't do well at all. And this green one here is a true random learner. So it basically picks a function uniformly at random. If you pick a function uniformly at random, you're not going to do better than random chance, and so you do badly. It's not surprising. So this is a nice illustration of what this does. I can look at this in more detail here. So here I've got a simple function. Here's the generalization error and the Lempel-Ziv complexity. And here you see this little cloud of blue dots are what the neural network does on different initializations. So I pick an initialization at random, I train it with SGD, I see what it until zero training error, then I see how well it predicts. It doesn't always do exactly the same, it's a stochastic model. But you get this little cloud around here. Actually, the most likely function is actually the simplest function. For a slightly more complex one, it does this, the chaotic one does this, the random one does that. And then for a very complex function, it gets worse and worse and worse eventually. And you get these clouds, these clouds of what these things do. So that's basically, so I've done a control experiment by breaking the simplicity bias. And I show if I do that, I get dramatically worse generalization performance. And the question is, why is that true? there's a number of different ways you can work that out. But since this is talk about AIT, I'm going to give it like an AIT 101 explanation of this. So what I really ought to think about is how likely am I to get a function of a certain complexity? Okay. Well, what we know from some accounting arguments is that the number of functions of complexity k grows as 2 to the plus k, right? So if I have of functions of that are length 128, then the vast majority of those functions, if I pick them at random, will have complexity close to 128. So here, this red curve here is an ideal compressor, which basically compresses everything to the shortest possible string. And so there's the maximum complexity then is 128, and the 50% of the strings are going to be between have either 128 or 127 as their shortest description. Then 50% will be 126 and all the way down. So by the time I get down to the complexities down here, there's extremely rare strings. Okay. The green thing I'm showing you is our Lempel-Ziff compressor, which is not an ideal compressor. We spent quite a bit of time looking at different complexity measures. You see what this compressor does is it kind of it sloshes out the complex strings a little bit, but it does well in its scaling for the simple strings. And the point is extremely simple. The coding theorem predicts a 2 to the minus k likelihood of an individual function. The number of functions grows as 2 to the plus k, and these two cancel each other, right? And in a normal network, you get a relatively flat distribution of complexity. So you're basically equally likely to get a high complexity or low complexity function. That's just a kind of hand waving argument, right? 2 to the minus k, 2 to the plus k, they cancel each other. If you have a neural network, I guess chaotic one, which is simplicity biased, but not quite 2 to the minus k, but 2 to the minus 0.9 k, for example, right? Then it can't overcome the exponential growth of functions, 2 to the plus k, and it basically is highly biased towards complex functions. Go ahead. So in this one here, on the left one, yeah? No, on the right. The right. Yeah, so you randomly sample parameters, and you look what it does, measures complexity, and bin it. Yeah. The kit. No. So there's a whole kind of subfield of this where you can show that if you use certain activation functions and you use wide Gaussians to train, you get into this chaotic regime. In the chaotic regime, you basically lose memory of your input as you go to the output. Yeah, exactly. And that generates effectively what I show here is that effectively generates something like, it's not quite random sampling over parameters, but as you get further chaotic, random sampling over functions, but in the limit of fully into the chaotic regime, you'll be effectively uniformly sampling over functions. And in between, you're doing something in between, but you're no longer simplicity biased in the right way. And so if you think about it, what is bias variance trade-off doing to you? It's basically telling you, there's a whole lot of functions that can fit your data. If you don't have some kind of regularization, you're going to pick one of those with high probability, and the vast majority of those won't generalize well. That's the argument. And this is just a simple 101 way of illustrating what these neural networks do. So this is all with training. Yeah, these are all training SGD. I see where your question is going. I'll get to you in a minute. I've skipped over something that also confuses people, which is that I've jumped back and forth between things that I trained with SGD and things where I only look at the prior. And I'm claiming just looking at the prior tells me what the trained thing does, right? Yeah, and this is controversial, but I think it shouldn't be, and I'll tell you why in a minute. And here I show you just another little game we played. So you can also, instead of running a neural network, you can do an Occam's razor learner. So I give it a function of certain complexity, in this case, a simple function, and then I pick functions at random, but I cut off my complexity by functions. I only allow functions up to a certain complexity. That's my green one. And then if I make my complexity larger and larger, I predict less and less well. That's what you see here. This neural network, I can do that as well. If I take a neural network and I force it to make the simplest function that fits the data, it's slightly better than the full neural network, but not much better. So neural network has a kind of statistical Occam's razor. It's not a perfect one, but it's pretty good. And you can also derive bounds. It's a pack bound, pack base bound. You can do a bunch of bounds. It's all simple and nice to do. So their question was, hold on, why should the parameter function map explain neural network outcomes? Because I don't train a neural network by randomly picking parameters. I train it by giving it some data and then running SGD, which is running on some very complicated high-dimensional loss landscape, looking for something like a minimum. And one of the dominant hypotheses in the field is that some property of SGD is what causes is what regularizes it. And this is a very common. I've been told this many, many times. I don't believe it, but this is a common hypothesis. And here I have a little example of how you might think about this empirically, at least. I mean, SGD is a very complicated problem. High-dimensional optimization problems are very complicated. It's hard to know what's actually happening. People make all kinds of proofs. I don't believe most of the stuff that actually is relevant to SGD in real life. But you can do experiments. And so here's an experiment where on the y-axis, I calculate how likely I get a certain function given training set. This is for MNIST. And on the y-axis, I do SGD. What I've done here is I've trained on 10,000 images. I test on just 100. I've binarized it, so it's even simpler. So 0 to 5 are 0, 0 to 4 are 0, and 5 to 9 are 1. Okay, so it's a very simple thing. So I've got 100, I've got 2 to the 100 possible functions. And then what I do is I train my neural network with SGD. I find, for example, that this function which has one error, so one of these images incorrect, number 35, my list of 100, for example, and that's the one that SGD keeps converging to. Right, it's the highest probability. It comes here in this case, a fairly high percentage of the time. And the y-axis, I have a Gaussian process, so it's the infinite width limit, but it's perfectly Bayesian, so it's basically equivalent to randomly sampling over parameters. And you find that it basically gets the same function with high probability. So a system with no SGD and a system with SGD are giving you essentially the same functions with some noise around it, right? But the first order, that's what's happening. And so the claim that we make is basically, you're in a very high dimensional space. You've got lots and lots of potential minima. Each of the minima is basically zero training error, of which there are an extremely large number of functions that will give you zero training error. The question is, why does SGD converge on a function that gives you good generalization? It's because that function has a large volume in the space of parameters. And if you have a large volume, you've got a large basin of attraction. So you're going to fall into that basin of attraction much more likely than you're going to fall into the basins of attractions of functions that have smaller volumes. And since those volumes are proportional to the complexities of the outputs, you're much more likely to get simple functions rather than complex functions. That's the claim, which I've washed over some details, but that's the claim why that works. And just to show you that this same idea works for, you can do it for MNIST. So the minute you look at more complicated data sets, it's harder to measure the complexity carefully. You can't really fix your target problem the same way because the target is just learn MNIST. But you can show here, for example, I show you what the neural network learns for a normal one and for one that is more chaotic. And you see for the one that's normal, you get low error and low, this is critical sample ratio. It's another way of calculating complexities. I won't tell you what it is, but it's basically a complexity measure. And this is for one that's very chaotic, which generalizes less well and also is considerably more complex. This is the prior for the normal one, the prior for the more complex one. So as you go into this chaotic regime, you see that neural networks go towards more complex functions and their generalization performance deteriorates. So our claim here really is that neural networks are intrinsically biased towards simple functions, Kolmogorov simple functions, so functions that have simple descriptions in some way or the other. And in fact, that's good if my things I'm trying to learn are also simple. Great. For the Boolean system, what's nice is I can actually control the complexity. I know what the complexity is. I know exactly what I'm doing. For something like MNIST, what is the complexity of this thing? It's the function that takes these pixels, 784 of them, okay, and turns them into labels. Okay, that's like some complicated thing. I don't know exactly what the complexity is. I don't know how to measure it, but I do know this is relatively simple. So I can look at things like the dimension of these images. They're in a 784 dimensional space, but the dimension, if you use various dimension measures, the kind of same things you do in chaos theory, for example, you show that dimensions are very slow, like 14, 15, 10, or the typical dimensions of the images. So these are low dimensional objects in a high dimensional space. And I'm going to claim that basically neural networks, when they work well, are effectively learning low dimensional structures in very high dimensional spaces, structures that are somehow k-simple, Kolmogorov simple. So what I said in this first little bit is, why don't heavily over-parameterized neural networks over the data? That's a Ryman's question from many, many years ago. I think we don't still completely understand. We need a new statistical learning theory of over-parameterized systems. But one key thing is that they have to have an inductive bias. That's actually, we know they have an inductive bias because they can fit any function, so they have to be biased towards certain ones. And bias towards simplicity is definitely one of the inductive biases that neural networks have. And without that bias, you're going to get overwhelmed by the fact that the number of complex functions grows exponentially. Then the argument is that that bias is present in the prior, and that's the dominant source of the bias. And stochastic gradient descent or other ways that you try to train your network may or may not aid generalization, but probably not answer the first order question, which is why don't overparameterized things fit? And there's some other things about no free lunch you can think about. The second question, given a neural network that works well, how could we get better generalization? This is what you do as a practitioner, super important. I think for theorists to understand this, we're going to first need a proper theory of what's happening in an overparameterized regime. What's happening in an overparameterized regime is very different from the stuff you have in your textbooks on statistical learning theory, and it's going to have to include something. It's going to be a story about inductive bias, and this AIT bias is one of the key stories that I think you're going to have to need to explain why neural networks generalize. Any questions? Yeah, so there are, so I skipped over it, but we use something here called critical sample ratio, which is something basically something that longer. It comes from this paper here, if you want to look it up, or Pitel. So there are analogs. They're just harder to work out what they are. But intuitively, one of the arguments is that you can take these different analogs, they should be proportional to each other. Definitely on this kind of logarithmic scale, they should be. And that's what you're basically looking at. Yeah? Yeah. Yeah. So you can, each of these dots is the probability that the GP will generate that function. It's not the mean, it's the probability. The GP gives you probability distribution, right? So for each function, you can tell the GP will give you different probabilities. Right? And so these are probabilities on the y-axis, the probability the GP tells you for these functions. On the x-axis is what you get upon repeated running of SGD. Yeah. So this is the I get most of my papers I have been turned down on the grounds that we all know SGD does it. You can't possibly be right. And it drives me batty because I can take a GP, an NGP, and get on all these data sets more or less the same generalization performance that I get with an SGD trained neural network. Right. So there's no way the SGD, if I can get the same results with a system that knows nothing about SGD as I can get with a system that knows SGD, then clearly SGD is not the source of the inductive bias. It can't be. Right? So although it's trivial, I think, to show that out, it's so dominant in the fields that people keep repeating it, but it's wrong. Well, Jeremy can explain a bit more about this, but well, she will converge that posterior. That's great. And the NGP solves the overparameterization problem. Yeah. So does the SGD trained neural network. Yeah. And they do it exactly the same way. Ergo, if you can do it without SGD and with SGD, SGD is not the source.
This is what I'm saying, though. So I suppose there's some feature of SGD, right? But any operator given with that has some like magic property.
Yeah, no, so the SGD is magic argument is the one I get all the time, and I think it's nonsense, right? Fine, you find somebody has to give me what the magic is, right?
I'm not saying that it is. I'm
just
suggesting that the chart might have rather than taking the N width to infinity limit as the y-axis, use a different optimization.
Yeah, so we've done that as well for small. For the Boolean system, you can do just direct sampling, and it gets the same result. I just show this one because it's for MNIST. But if it forces you more to do it per, you can do other optimization method. Totally works. Same thing. Yeah.
It's possible that in the width to infinity limit, whatever the magic is remains, and so the magic is actually still in the GP.
Maybe there's some magic to it. Maybe this is I get this again and again. Yeah, you say this, but SGD might be magic because we all believe it's true. Maybe, but show me the magic. That's all I'm saying. So if there's a magic result, show me.
And there are, say, this is the first order argument, right? The second order, for sure, there are things that SGD does that help with generalization. It minimizes norms. There's all kinds of things that are true, right? I'm just telling you for Bryman's question, SGD is not the answer. Yeah. All right. That's the main thing I want to talk about with machine learning. I'm going to just zip over the evolution because I've lost track of time. Super, that's great. So actually, there's a similar kind of problem to the overparameterization problem in evolution as there is in neural networks. And I'm telling you this in the opposite order in which I came into this problem, which is basically that, so Charles Darwin famously came up with natural selection and variation, right? Variation means your offspring are different from their parents, and natural selection means you select on the ones that have the highest reproductive fitness, so the ones most likely to have a viable offspring. Famously, Darwin didn't know about Mendel, and when Mendelian genetics was rediscovered by Hugo de Vries, he suddenly thought, oh gosh, evolution goes in these discrete steps. Okay, so variation happens in discrete steps, and de Vries famously said natural selection may explain the survival of the fittest, but it doesn't explain the arrival of the fittest. So the question is, in order for natural selection to work, it's got to first have something that it works with. And that's kind of, so I started working on molecular systems because they're easy. And just a little back thing for you guys is probably obvious, but not for many biologists. These spaces that evolution works in are extraordinarily high. So DNA has 4 bases, RNA has 4 bases, proteins have 20 bases. If you made every length 58 protein, it would weigh more than the mass of the observable universe. Okay, that's a very short peptide. The mean length in your body is about 476. So these are spaces that are unsearchably big. Okay, I'm going to look at RNAs. RNAs can also act. They're molecules that can be messengers, but they can also act catalytically, or they can have they can act biologically in structure. And so there, I think every length 126 RNA would weigh more than the mass of the observable universe. One answer to this question is that these spaces are very big. So here's a space of all possible RNA structure, for example. Here's a space of all possible things that RNA can do, which is smaller, right? So some sequences are similar to each other and give you the same output. That's not too similar to the story I just told you about the neural network, right? So many different parameters will give you the same function. There's a redundancy in there. There's a redundancy here, right? Many different sequences will give you the same RNA structure, for example. Many different parameters give you the same function, same story. Most of evolutionary theories worry about the second thing. I've got a bunch of different phenotypes. Which one's going to be selected in the population? How does that stratify, etc.? I'm going to look at this first question, which is, if I just randomly pick sequences, how likely am I to get a certain structure? I got interested in this from this experiment by Jack Shostak, who's a Nobel Prize winner at Harvard, who did in vitro lab evolution experiments. So he took the same RNA, and he looked for a certain enzymatic activity. So enzymes is something that's very important for catalytic behavior in cells, is how your cells control life and how they control everything. And look for a particular enzyme, and what he found was every time he ran these experiments in his lab with tiny little test tubes of length 55 RNAs, he always found this particular shape when he selected on a particular kind of enzymatic activity. So I calculated how much material he used. It's about maybe 1/100 of a gram. If I made every length 55 RNA, it weighed 10 to 10 kilos. Okay, and so how can we find the same thing every time? It's like putting your hand into a giant haystack and grabbing a needle every single time. So why does Shostak find the same structure every time? He talks about this convergent evolution. So evolution in this hyper astronomically large space finds the same thing again and again. The reason I pick RNA is because there's a well-developed set of computational tools that will take you a sequence like this one and fold it for a lowest free energy state, which is this one. And then what you, which is basically this, and this tells you who binds to whom. So I can take, like this is a short one, like 15. I can write out, this is a little structure that I fold to a hairpin structure. I can abstract that out and say the first 4 bonds, the next 5 don't bond, the next 4 they bond back like that. I can draw that as this little simple structures. And so if you take every length 15 RNA, and you randomly pick sequences and you fold them, what you find is that this one gets 12 million folding to it. This one only gets 2 folding to it. By the way, this is one of those low K, low P structures, low complexity, low probability. And it ranks over many orders of magnitude. And of course, this is because these can have the same simplicity bias. So here I take like 55 RNAs. Here's the simplest one, which is no bonds. That's its probability that it appears on random sampling. Here's a more complicated one. That's probability or log probability. And you see that the simplest one is exponentially more likely to appear than the complex structure, right? So nature has this inbuilt Occam's razor towards simple structures, is the claim. And what's nice about these these systems that you can easily write down a kind of bit string version of them and compress them. And so if I, instead of plotting versus complexity, I plot just versus rank, then what you see is these green ones are the ones that are used in nature. There's 504 sequences that we found of this length that are used in nature, in natural organisms. They're all the ones that have very many sequence mapping to them. They're all the ones in this thing at this low complexity. And that Hermann Reibersheim that Schustak found is one of them. We think there's actually 10 to the 13 possible structures at this level. Okay. And nature only uses this incredibly tiny fraction of them. And so why does Schustak keep finding this needle in the haystack? Well, it's because it's one of these things that is extremely common. And so it's kind of interesting, just like the neural network only uses a small subset of the functions that it could do, this RNA system only uses a small subset of functions that it can do. And you can do this in more detail. You can abstract these out a little bit. And here I show you, here's length 126 abstracted structures. We think there's 10 to the 12 possible structures at this level. Only 63 are shown in nature. And then I rank them by how likely they appear at random. And basically you can see that they're the ones found in nature are almost step by step, all the ones I find by random sampling. So all the complexity of RNA that's used in nature, okay, can be found at this level by maybe 1 million random samples. It's very surprising. There are trillion possibilities, and nature only uses a tiny fraction of them, and that fraction that uses are the ones that are Kolmogorov simple. That's kind of interesting. And then the last thing I'm going to do in the last 5 minutes is show you another example of this. So that was the RNA structures. The only point is that they're easily compressible. Nature likes highly compressible structures. There's actually the variation is built in to do that. And maybe I should say one last thing I didn't say is, like this is like 55. I've on the first 183. There's 10 million of them. The other ones are the ones found in nature. You can see that after this, none of them are found anymore. And the argument for that is almost certainly not natural selection. The argument for that is that these are the ones that appear with highest probability. They're the easiest ones to find. And the ones that are easiest to find are the ones that nature uses. And this is very surprising for a hardcore adaptationist Darwinian, because I'm explaining something about the way the world is, ignoring natural selection entirely. I believe natural selection is definitely at work in this case. It can only work with that which it's given. And that's a small subset of all the possibilities. And the last one I want to show you, which is kind of fun, is this is a little movie by Keiji Namba of the bacterial flagellum motor. So bacteria have these little motors, they spin at 100,000 RPM, absolutely exquisite little things. If I showed you one, you imagine it would be built in a factory. Okay, I could hold one in my hand. But these things are made by self-assembly. So the cell makes little proteins, they float around, and they spontaneously form into this well-defined three-dimensional shape. Half the proteins in your body are in some kind of complex, or some kind of, they're dimers, trimers, tetramers. And they're produced by an evolutionary process that just randomly picks sides and mutates them and has them synchronous. They're thinking Lego blocks, putting some glue on it, shaking it, and out comes a fully formed train. This is what nature does all the time. How does this work? So we've come up with a simplified model of these proteins. Here's an octamer. We're going to simplify this as a little two-dimensional shape. So these are little particles here with little sides on them. And this is called polyominoes, like dominoes is 2, triminoes is 3. It's a branch recreation mathematics. This is kind of fun. So I'm going to make a set of rules that say, like here, A binds to C. So I put A down 1 found first, then C, A binds to C. 3 has C on it, so 3 is going to bind here. And if you follow the rest of the rules, you're always going to make this structure. You want to always make the same structure each time, because otherwise, imagine that motor that self-assembles into a slightly different structure, it won't work. So you need to self-assemble into a really well-defined structure. So I can now turn this question around. If I want to make this structure, can you give me a set of rules that will make it? Well, you probably could. You can use graph theory to solve it. But evolution doesn't know about graph theory. It just randomly picks patches and mutates them and adds and subtracts them. So my student Ian said to me one day, I'm going to try this. I'm going to make 16 mers. Well, I'm not going to tell you which 16 mer I'm going to make. I'm just going to say I'm going to run an evolutionary algorithm that has maximum fitness for a 16 mer. So there's 13 million 16 mers. So you might think I get each 16 mer with equal probability, because I haven't told you which one to make. But when he ran the simulations, he found that this small set of 21 appear 50% of the time. Okay, so they're extremely highly biased. And what's kind of cool about them is that they're highly symmetric. So these dark blue ones you see here are the only 5 16-mers that have D4 symmetry. So I can rotate them and flip them and they're themselves. And there are only 5 in the 13 million. And yet they're the ones, I find them about 30% of the time if I'm running an evolutionary algorithm. That's kind of cool. So evolution is basically biased towards high symmetry structures. And here I show a P versus K plot again of these things, not surprising. And here I show the green one, the natural ones. And it's not surprising. A symmetric structure doesn't need very much information to describe it, right? I take something and repeat it n times. So if I'm running an algorithmic process, if I'm randomly sampling in a space of algorithms, I'm much more likely to get a symmetric structure than a non-symmetric structure, even though there's only a small number of them. That's the intuition, which you can formalize in lots of different ways. And indeed, if you look at nature, here's 6-mers, for example, hexamers. These are low complexity hexamers, and this is their frequency, they're very frequent. These are high complexity hexamers, they're very rare. And the argument that we make here is basically this kind of coding theorem-like bias towards simplicity is so strong that it explains why you see so much symmetry in natural proteins.
Yeah, so I'm also a little bit skeptical about this in that the symmetry is the visual symmetry, right? It's something that is that it looks in the plasticity and visual cortex. But there are lots of other symmetries, like super symmetric in 4 dimensions, or follow the digital pi stuff that we cannot see.
Yeah.
That you need to homomorphic complexity would be equally simple. So why do these symmetries, these visual symmetries come out?
So that's a good question. So one question would be why, so what fraction of this, so actually take a step back. If you look at 2-dimensional symmetries, you can point, there's a very limited, you can ignore your own cortex and just mathematically count the size of the groups. And there's a limited number of these 2-dimensional symmetries for 16-mers. I think there's 5 different ones or 6 different ones, I remember right. And that's it. So let's ignore our visual cortex. Those are the ones that mathematically you can get, and you're going to be biased towards these. If you map them into 4 dimensions, you can make other symmetries, right? But these things live in low dimension space.
So it's the other way around. We have a visual cortex.
Yeah. So whether our visual cortex, so this is a very deep question, right? Does our visual cortex, do we have biases in our learning, in our minds? For sure we do, right? Are those biases towards certain kind of simplicity? For sure it's true. Is that why we see patterns in nature? That's also true, right? But is that what we're seeing here? I think no. The other question you asked, which I think is a very deep question, I'm just going to thank everybody for all the hard. The deep question is, so we're doing this kind of crude, physicist level, not mathematically completely tied down versions of the coding theorem for non-UTMs. Probably closer to MDL than anything else. And so one of the things we're assuming is that things like pi, which are algorithmically simple but that will break any compressor, are not happening with high probability in these systems, right? And if they would be, then we wouldn't be able to, our complexity measure wouldn't miss it, right? And so we classify these as complex, and they shouldn't be. And so the fact that it works is an a posteriori way of saying that must not be very important. You can ask Marcus why, whether this is deep property of the world, right? But
just to comment on that, I mean, I mentioned earlier, Lieber's having Paul Battaglia, who's obviously famous in this field. He's mentioned explicitly that, why is it that these everyday compressors are working as if they were, more complexity? It's probably because he said because the systems we're looking at are not actually universal Turing machines themselves. So, like, you might be saying, why is it that some crazy symmetries, let's say, pseudo-random symmetry is not being discussed or worked with on? It's probably because the complexity measure can't measure that, and the system itself is maybe closer to a finite state transducer or some other kind of limited computational system. So it's not making those kind of crazy patterns, and our measure cannot detect them. But that's not a problem because they're both really the same thing.
And in fact, you could, this particular tile model, what you can prove is not Turing universal. There are Turing universal tiling models, but you need a few other extra things in it to make it Turing universal. The RNA system, we know, is a context-free grammar effectively, so it's definitely lower on the Chomsky hierarchy. whether that is, these are the difficult questions. So, but I think it's a deep question. Right? So the two questions are, are we biased towards certain things for sure? Right?
But also that's like related to what Marcus was saying earlier. Why are we using Kolmogorov complexity? But perhaps Kolmogorov complexity proper is not the actual thing that we want, because it provides a theoretical framework that you can actually ask questions and compute things and then try to apply them. Whereas, that's for me why AIT is also interesting. Even if you can't actually compute it, or maybe some aspects are not going to be appearing in the real world, at least it gives you some way to think about these problems, come up with some kind of sort of approximation.
These are various people that helped us. These two students are here today. Thank you for me, and obviously Kamal in the machine learning, and these people did the evolution work because Kamal's on both. And so thank you for your time and attention. Any more questions?
Yeah. Yes.
About the variance of the neural network as it increases and goes towards chaotic. What happens when you just shrink it?
It goes to an ordered regime,
right,
where actually we don't have time learning.
Okay.
Yeah, but it doesn't. As far as we know, it doesn't.
Does it just limit the kind of functions that?
Yeah, if you make it too small. Okay, so this is the interesting thing. Is this just if you're running SGD, it shouldn't matter where you start. You should eventually be able to reach whatever. Right? And if SGD, if you make it too wide, actually SGD has something we think is kind of a technical effect, which bounces the. If you make the initial variance too wide, the SGD actually bounces back to lower variance. And this is has to do, depends on which optimizer you use. So this gets a bit tricky and messy. But if you make it very, you basically lose expressivity initially. Yeah. But yeah, the basic point is that so we worked here in a regime where SGD and let's say Gaussian process or random sampling parameters give you very similar output. If you go to the very, very wide regime, you start seeing differences. So actually, the fact is, if you took the infinite depth, or almost infinite depth, almost infinite width, almost infinite initial width, so not the width of the layers, but the width of the initial parameterization, SGD might be able to, if you fiddle it right, be able to get you back to something that works. But there's some effects that do that. Yeah. So we have a 80-page paper on bounds. Yeah. There's huge generation bounds for deep learning, and actually, I think Jeremy, you've interacted with us a little bit on this and helped sharpen our thinking. So you can make a generalization bound based on pack. So there's a thing called pack base. Yeah. It gets hugely simplified in these kinds of situations. So the pack base bound, basically, the bound on your error is given by the marginal log of the marginal likelihood. Yeah. Right, and the marginal likelihood, if you think about it, is basically the probability that if I randomly sample parameters, that my neural network gives me zero error on the training set. That's the marginal likelihood of a training set. So what it basically tells you is the error is bounded rigorously in this case by the probability that you find a training set upon random sampling up to some delta constants. And it works. Here's how you see it works as I have a label corruption, right? My generalization error goes up and my bound goes up. Right? If I do things like here's for a whole bunch of different things, I have a learning curve. So how many I change the size of my training set. This is for all these different res nets, dense nets, mobile nets, fully connected network. And you can see how there's a famously these neural networks show a kind of power law like distribution of the error as in the learning curves, and we can reproduce that based on these bounds. So these bounds are not, they're not perfect, but they are bounds. The top, yeah. Top's the bound. Yeah, it's non-vacuous, yeah. So these bounds are non-vacuous. In fact, for various reasons, the back base bound is trivially non-vacuous, but it's non-vacuous, and it shows the right scaling. And as far as we know, there's no other bound except Jeremy's that does this. Yeah, but then there's a big literature on bounds. But I think it's a very big literature and bounds are very interesting. But this I think is one of the best performing bounds, and it works extremely well. And
I
I had a question too. Well, Hello? Wait, what happened? I don't see anyone anymore. Or